As we've mentioned on this channel before, the hallmark of a good theory is its ability to make predictions.
I'm guessing he'll just ignore the massive amounts of failed predictions in this video.
A hypothesis must have predictions, must be accurate in explaining the world around us. Okay, let's look at the Bible, for example. The Bible tells us clearly that it is the history book of the universe. And yet, why is the known genetic data so absolutely consistent with a biblical based model? It's really far too close for comfort for the evolutionists. We've made future testable predictions, for example, on mutation rates. We've made testable predictions on mutation rates in the Khoisan peoples, on DNA function. We've made flood geology related predictions, prediction after prediction. When we actually look at the genetic data, for example, let's make some retrodictions. Let's see if the Bible really is the history book of the universe. Biblical Adam and Eve predictions, for example. Well, you'd predict low genetic diversity. You'd predict one Y male ancestor, one mitochondrial DNA female ancestor, the fact that most genes come in two versions. If Eve was taken from Adam, Noah's flood predictions, for example, you'd predict that all people should be closely related. There's only a few mitochondrial DNA lines, the one Y chromosome line, evidence of rapid population growth. And at the end of the day, we actually see, based on the three main events in Scripture, specifically in Genesis, we can actually address these with modern genetics. These events, for example, the creation of Adam and Eve, the flood of Noah, the Tower of Babel. Let's look at the data. We now know that all human beings are actually profoundly similar genetically, and this points directly to Adam and Eve. If you only started with two people, that restricts the diversity today. Over deep time, any large population, of course, I hope Jackson understands this, will most certainly and undoubtedly accumulate enormous numbers of mutations, resulting in enormous amounts of genetic diversity. This is a serious problem for evolutionary theory because it is now absolutely clear that mankind has very limited genetic variation. While the observed low genetic diversity is a massive problem, of course, from the evolutionary perspective, the perspective and the fairy tale that Jackson here is pushing, this is most certainly expected and predicted, as I've said, from the biblical perspective. From the biblical model, we all come from just two people. This obviously means that limited diversity is easy to explain. The Bible predicted this. This means the death and demise of the evolutionary bottleneck theory and the evolutionary out of Africa theory. Now, what they're going to say, they're going to resort to a near extinction event in order to reduce homogeneity, but it would cause permanent and severe genetic damage. For example, enormous numbers of deleterious mutations will undoubtedly go to fixation. But based on this low genetic diversity, based on the fact that every single human being in the world is very similar, they have to resort to what they do best, a rescue device. Jackson Weed here presents some predictions. Why doesn't he talk about all the failed predictions, all the inconsistencies, all the contradictory evidence? They cherry pick, selective and preferential treatment of data, of course. This apparent bottleneck, this near extinction event, it is just that. It is a rescue device. How could such a tiny, nearly extinct, genetically compromised population suddenly explode into all parts of the planet and seizing dominion over the world? Because this hypothetical, it's all storytelling, it's all hypotheses, unsupported hypotheses. And this apparent evolutionary bottleneck, although it might have been able to reduce overall human diversity, which is what they're trying to do to explain the data, you must understand, and Jackson here talks about post hoc explanations, rationalizations. Well, this is just a post hoc embellishment to the evolutionary fairy tale. It is not actually credible at all, because as I hope he understands, small bottleneck populations have extreme problems. For example, there are approximately 10,000 cheetahs in the world today. Look it up. And conservationists, people who are worried about this, they acknowledge that the cheetah is already showing serious signs of inbreeding and genetic decline. Genetic diversity has eroded, of course, based on inbreeding. The evolutionary bottleneck hypothesis that they pull to explain this data, but the Bible already predicted it thousands of years ago, involving this extended near extinction event, which is obviously associated with severe inbreeding, as I've demonstrated here, it's not even remotely plausible or feasible. So from the evolutionary perspective, human genetic homogeneity still remains, regardless of what they want to tell you, regardless of the rescue device and story that they pull, remains a very serious theoretical problem. But according to us, according to the biblical based model and biblical perspective, there is absolutely no problem with a relatively homogeneous human population. We start with just 2 people, of course. And this population bottleneck, according to the Bible, is benign. And then 10 generations later, we see a second single generation bottleneck of just 8 people occurred at the time of Noah. And both bottlenecks, of course, read Genesis. Look at the data. They were both very brief and then followed by explosive growth. And in both of these cases, there would have been almost no previously accumulated mutations. We see the accumulation of low impact, near neutral deleterious mutations. We see genetic entropy. Take that back to a point of least genetic entropy, that would be a point of creation. At this point, there wouldn't be that accumulation of mutations and hence no inbreeding effects, of course. I can't believe evolutionists still bring up that objection. It's a straw man. So very limited human genetic diversity is a massive, it's pretty much a fatal problem for evolutionary theory, and it leads to unrestrained storytelling. That's what they do. The evolution story needs to be revised almost annually. Yet limited human genetic diversity is very obviously compatible with the biblical perspective and does not actually require any far-fetched mental inventions. And I mean, we can go on and on and on about these predictions. We can go on and on and on. I mean, for example, let's look at Noah's flood. This story tells us that all people should be closely related. It tells us there should be only a few mitochondrial DNA lines as Shem, Ham, and Japheth's wives, and only one Y chromosome since it's only Noah. His 3 sons would have inherited his Y chromosome. And we should have significant evidence of rapid population growth. And it turns out this too is exactly what we find in modern genetics. Guess what? This did not all have to be true. The Tower of Babel account, for example. We have found a single dispersal of all people in the relatively recent past who traveled in these small little people groups into uninhabited territory through the Middle East. Too close for comfort for evolutionists. They call it the Out of Africa story. As I've shown and demonstrated, that's dead. But these are all biblical-based predictions.
We've talked a lot on this channel about how evolution is used to understand the past, but today we're going to talk about how evolution can be used to predict the future. If the evolution of all organisms from a set of common ancestors is true, then we should be able to test that. An example of a modern prediction of evolution is one we have discussed at length, the chromosome 2 fusion, including all its details. Using these frequencies, we can generate a graph like this, showing the signature of mutations. If mutations were also the cause of interspecies genetic differences, then we would predict a similar spectrum graph when counting up the different types of nucleotide differences between humans and chimps. And we do. This makes no sense under a non-evolutionary model. Under creationism, it implies that a creator created interspecies DNA differences that just so happen to look exactly as though they had occurred by the same natural processes that give rise to within-species differences. It also makes no sense under a non-evolutionary model why genetic sequences for homologous proteins converge the further you go back in time, which was, by the way, also predicted under evolution.
Jackson Wheat here uses a lot of arguments that assumes the majority of our genome is actually non-functional or simply based on evolutionary leftovers. Evolutionists like Jackson here, they assume that all DNA differences are the result of mutations over time, of course, and based on our model of pre-existing functional heterozygosity, we would actually assume and predict that the majority of our genome is created diversity, that God encoded these functional DNA differences from the start. And what's funny is we now know that the junk DNA era has been completely overturned. For example, this human chromosomal 2 fusion that Jackson here says arose through a fusion of 2 ape-like chromosomes, that alleged fusion, that alleged site where the fusion supposedly took place actually represents a highly organized functional gene. And as you can see, there's significant lack of evidence for a cryptic centromere site. And we're going to get more into these various arguments that he uses on endogenous retroviruses, specific pseudogenes, things that assume evolutionary leftovers, and we're going to get more into that. But in regards to the chromosome 2 fusion, he fails to mention that Dr. Jeffrey Tompkins wrote a extensive technical paper in February the 8th, 2017 titled Debunking the Debunkers. It's a response to criticism and regarding the refutation of the human chromosome 2 fusion paper that he originally put out back in 2013. And in this paper, you can read for yourself, you see a ton of genomic data after data, evidence after evidence that clearly shows that the alleged fusion site really is a promoter in this DDX11L2 gene, and that the activity, contrary to Jackson Wheat and other evolutionists that have attempted to rebut this incoming evidence, is neither nominal or spurious. And there's so much data in this paper and coming about that you can read that shows that the alleged cryptic centromere site has actually no homologous chromosomal matches in the chimpanzee genome. And I quote word for word here from this paper titled Debunking the Debunkers, that it is also shown the site is too small to represent a centromere, and that interstitial alphoid repeat regions are common on many human chromosomes, including chromosome 2 that has 6 different alphoid-rich repeat regions outside the centromere. However, the most negating evidence against the cryptic centromere site is that it is completely situated inside the actively expressed protein coding gene. In fact, Dr. Tompkins says in this paper, sections of it extend across both intron and exon regions according to a line transcript, and the alleged cryptic centromere is clearly a functional part of a protein coding gene and not the remnant of a centromere. Now, according to the pre-existing heterozygosity model, the created heterozygosity model, we would predict that the vast majority of our genome is functional, of course. And Jackson here uses some of these shared genetic mistakes, these pseudogenes, to prove evolution. For example, the broken remnants of an egg yolk gene. And these pseudogenes are now known to be functional DNA elements and not actually mistakes. The junk DNA era, the junk DNA paradigm, it is dead. It's been overturned, and the fact is we understand very little of the DNA language. We're dealing with a very foreign language here, and we've only just begun to discover and uncover just how functional the human genome really is. It's mind-boggling how many experiments would actually be necessary to conclude with any amount of confidence that a certain set of DNA sequences has absolutely no function at all. And there's preliminary biochemical evidence for function It doesn't just exist for the known function, the known functional pseudogenes, as I pointed out, but also for at least 80% of all the pseudogenes in humans, for example. And given all the data that we know, the evidence that we know, it's obvious that the other 20% may still yet to be discovered to be functional in some human tissue, or even under some physiological condition that has yet been studied extensively. I mean, many non-coding RNA genes, such as these pseudogenes, they're only expressed under certain conditions. So the fact that he uses a lot of this evidence that assumes mutations are the source of all variety, that assumes that the majority of our genome is junk, and a lot of them will say, oh, we never predicted junk, but you can talk to scientists. I've debated scientists, evolutionists. They say that the genome can be no more than 20% functional. Well, we're predicting that the vast majority of our genome is functional, and that's exactly what we're seeing, because the scientists, the findings, the influx of scientific papers are finding that numerous of these so-called fossil sequences, they're not pseudo after all, and these genes are absolutely necessary and required to sustain healthy life processes in the cell. You can look at some that have been overturned. For example, the beta globin pseudogene, that has been completely overturned. The evolutionists used to say that this pseudogene is just a useless genomic fossil, according to their own evolutionary predictions. But this gene, we now know, is a highly functional, and conveniently, it's an integrated feature of the human genome, and it's actually essential. This supposed broken remnant of a ancient egg yolk gene or chicken gene, there's no evidence for this remnant, that it even exists, and it's instead this fragment appears to be a functional DNA element. Junk DNA era, it's dead. I mean, they'll say the gene order along chromosomes has no function, and apparently and consequently this shared gene order demonstrates common ancestry, but the gene order along chromosomes does indeed perform a function. Apparently, Jackson Weed here will say that, humans are genetically closer to apes than to other, say, animal species, for example, using whatever type of gene sequencing, whatever type of phylogenetic analysis. And he says that it demonstrates common ancestry, but these relative hierarchies, they're actually characteristic of design, because our model does actually predict design. Of course, if nested hierarchies, the classification of life, for example, can be compared to classification of modes of transportation on how different companies manufacture cars based on their similarities forming these hierarchies. Humans, pigs, rats, and ducks, they're all land-dwelling. Even though they are separate kinds according to our model, since they are all land-dwelling, they can share similar nested hierarchies or design. It doesn't take much reflection at all to see how this model, the biblical creation model, the young earth creation model, predicts relative genetic hierarchies, and these are actually found in nature. For example, the God of the Bible, the one we are arguing for, he of course designed the entire universe, and this would actually consist of all the various kinds of biological life that actually live in it. We would then expect and predict to find that this life that God created fits a design pattern. So of course, if we as humans, according to the Bible, God created us in his image, we should be able to get a clear idea for what kinds of design patterns our creator God might have used. And we can get a sense for this and an idea for this by looking and studying and examining the patterns that actually result from human designs. And what we actually see is that means of transportation, they naturally fall into this Linnaean style categories. And Jackson Weed here, he uses examples of transitional forms, but as we know, many products of human design also evidently span two very different categories. I hope even he can agree with me that boats and tanks fall into entirely different classes. And Dr. Stefan Frello used a lot of this evidence as well. I debated him, and I would recommend going to watch it and seeing thorough refutations of these same tired arguments. But boats and tanks, they fall into two entirely different classes, yet there exist vehicles that actually blend the features of both these two extremely dissimilar means of transportation. Let's look at an amphibious assault vehicle, for example. This is resistant to classification. This would be the perfect example, according to the evolutionists, of a transitional form. But yet, what do we know? This vehicle is designed for the transitional environments between water and land. Basic rules of, say, efficiency and function, we predict function, will most certainly result in similar stages of manufacturing. If you want to look at nested hierarchies in DNA, well, it's obvious that we would find shared elements located in the blueprints of different manufacturing companies, say from across the entire planet. Let's take Toyota, let's take Chrysler, let's take Mercedes-Benz or BMW. Evidently, if the end products have similarities, the blueprints, of course, by necessity, have them as well. DNA hierarchies also fit the model of created heterozygosity and the model of biblical creation. So means of transportation, of course, they naturally fall and can be grouped into a hierarchical classification scheme. Like I said, these nested hierarchies, they're agnostic to the question of creation and evolution, as there exists competing explanation. And just to put it into perspective, I hope we can recognize and identify cars. Cars are obvious to anybody, even to a 5-year-old. And even though I hope we can agree every car is unique, all cars do evidently share a set of features. For example, a certain range of height from the ground, 4 doors, 4 wheels, lack of a cab bed, among more. Now, if we shrink the list of features, we can now bring in SUVs and trucks into this same group. I'm sure you can see exactly where I'm going with this. Trucks, as we know, are higher off the ground than cars, and since most trucks consist of a cab bed, the number of features shared among cars and trucks is less than those shared among just cars. And we can go on and on and on. We can then bring in motorcycles and so on. Technically, the more vehicles that we actually group and throw into our transportation classification system, the smaller the list of shared features becomes. And this is exactly like the Linnaean classification system. I mean, I hope we can all see that at some point the list of shared features among modes of transportation, as I am talking about here, becomes very, very small. Just think about bringing in and including boats and planes with land vehicles, for example. I mean, we don't even have wheels on the list at this point. This hierarchical pattern of life strongly suggests deliberate and intentional design. But as I've said before, we predict function. We look to function. And evolutionists themselves, they can make specific predictions on DNA function as we have done. But they haven't. And the trajectory strongly favors genome-wide functionality and the biblical creation model.
Cancer is a disease of genetic mutation, and it's these changes in the DNA sequence that really affect the behavior of a cell and then can lead to tumor formation. And so what we wanted to do in this project was really try to understand all of the genetic mutations that are driving pancreas cancer by looking at regions of the genome that most people tend to ignore. And so when you think of the human genome, you can liken it to a Broadway production where you have genes, and the proteins that they encode are the actors, the ones that we all really focus on. But it's important to remember that there are people backstage, the producers, directors, stage hands, costume designers who are critically important for making sure that the actors are on the stage when they should be, saying the right lines and singing the right songs. And so while most sequencing studies really focused on the genes, we really wanted to focus on the accessory regions of the DNA, the regulatory elements that tell genes when to turn on and when to turn off, really the stagehands of the genome. And so what we did is we developed a new algorithm in collaboration with Tyler Garvin and Mike Schatz here at Cold Spring Harbor and other scientists around the world called Gecko. And what this algorithm does is it looks through the genome and looks through all the mutations that people have in cancer and tries to really pinpoint those in those regulatory regions that are going to have a really great impact on the expression of genes that might be driving tumorigenesis. And so we found really a number of really exciting things when we wanted to apply Gecko to pancreas cancer. So first, we found these mutations in these regulatory regions do exist, and we could experimentally validate them back in the lab. Secondly, what we were able to find was a new prognostic marker that might tell patients more about their outcome when they are diagnosed with the disease. And then finally, we found patterns of these mutations suggesting that they're being acted on during tumor evolution near certain genes and near certain pathways that are critically important for cancer progression. And the implications of this is it really argues for whole genome sequencing of cancer, not just looking at the genes, but looking at the regions around the genes in the genome that tell the genes what to do to really give us a more full understanding of the genetics of disease.
Now, the other part of this that we want to talk about is something called chromosome fusion. Now, I want to tell you a few facts, and then I'm going to tell you the evolutionary story to account for these facts. So, it is a fact that humans have 23 pairs of chromosomes, and it is a fact that chimps have 24. Okay, so there's a difference. Now, if we came from a common ancestor, why do we have 23 and chimps have 24? They have to account for that somehow. Now, when they did initial studies of human DNA, this is before sequencing was really common and really quick, they would stain the DNA, the chromosomes, and they could see what are called banding patterns. And they noticed the human chromosome 2 was very similar to chimp chromosomes 12 and 13. And so just to show you that, so this is the real staining here, okay, the real chromosomes, and then they show it to you in a figure form. So human chromosome 2, they said, it's very similar to these two chromosomes, 12 and 13. All right, so they developed a story based off of that, and that story was, well, there must have been a fusion that occurred in the line leading to humans of two of those chromosomes, such so that we have 23 and chimps have 24. So here's how the story goes. In a common ancestor, there were 24 pairs of chromosomes. After they diverged, okay, so humans are going down this path to become humans, chimps are going down this path to become chimps, there was a fusion in the human line. Okay, two chromosomes fused together, so therefore humans have 23 pairs of chromosomes, and since all humans come from them, all humans have 23. In the chimp line, that fusion did not occur. So just like the ancestor, they have 24 pairs of chromosomes. Since all chimps come from them, they have 24, all chimps have 24 pairs of chromosomes. Now, this is not based on any evidential experience. This is just a story that they had to develop to explain this. It's not like you can go in the fossils and find this, okay? That's not reality. So this is just the story they've developed to explain it. And they would even go so far, some of them, as to say, it is definitive proof that humans and chimps shared a common ancestor. Now here's the thing, in science, we don't use the term definitive proof. If you're gonna use that, it's called a law. And you need to have a lot of things backing that up, backing that statement up, because that is a very strong statement. And I wanna share with you just some quotes from a few theistic evolutionists, again, professing Christians, but they believe in evolution in millions of years to kind of show that. The first is from Dr. Francis Collins, the head of the NIH. He said, the fusion that occurred as we evolved from the apes has left its DNA imprint here. It is very difficult to understand this observation without postulating a common ancestor. He said, how is it that human chromosome 2 looks so similar to chimps chromosomes 12 and 13 unless there was a fusion that occurred? I mean, how else can you explain that, is basically what he's saying. This next quote comes from Ken Miller, and he's a molecular biologist and theistic evolutionist, and he said, you know what? If we don't find it, if we don't find a fused chromosome in humans, evolution is wrong. We don't share a common ancestor. Whoa. Okay, so you're willing to bet it all on one piece of evidence. And so, and what's interesting is this quote comes from a YouTube video that someone posted of his lecture, and they labeled it "How to Shut Up Pesky Creationists." It has been viewed nearly 2 million times. Okay, so this is a very common thing that people will use to, so to speak, to say that, you know, we're wrong, chimps and humans definitely had a common ancestor. Now, although he says this, and he's made this statement actually several times, that he'd be willing to change his mind, basically, if it could be shown otherwise, I don't believe it. Because I can show that human chromosome 2 is not the result of a fusion. He has been presented with that evidence, and he has not changed his mind. Why? Because this isn't about the evidence. Okay, the evidence is clear. It's about a worldview, and not wanting to change that worldview, even in spite of the obvious, okay, wanting to cling to that. So let me summarize here what their supposed proof of evolution is. So they, or that we shared a common ancestor with the apes. So again, chromosomes 12 and 13 were in the ancestor, okay, and in the line leading to humans, a fusion occurred, and so now we have one that's known as chromosome 2 in human. This has gone so far, so to speak, that in chimps, they don't have chromosomes 12 and 13 anymore. They relabeled them 2A and 2B to represent the fact that they think it's a fusion that occurred in the human line. That's how strong this belief is. Now, if a fusion had occurred, what would we expect to see? There should be telltale signs of that. So you see these little dark blue caps on the ends of the chromosomes. Those are called telomeres. They're certain sequences of DNA. It kind of repeats itself over and over again. They serve as end caps to kind of protect the DNA. And interestingly enough, one of their jobs is to prevent fusion. So I'm kind of struggling how they were involved in a fusion when that's one of their jobs, is to prevent it. So we should see a bunch of telomere sequences right there. The other thing is this little red symbolic square here, that's a centromere. Every chromosome has one centromere, and it's important when the cells are dividing and the DNA material needs to be divided up between the two cells. There are certain sequences associated with those. So we should see one in the fused chromosome. It's not active anymore, but it should be there along with the real one. So that's what we should see. So we can sequence human chromosome 2, and we can look for those things. And that's what they've done. And so what did they find? Well, they're having trouble finding a lot of those telomeres in the middle. There's some sequences there that are sort of kind of like telomeres, but not nearly in the number they should be, okay, if they're truly telomeres. So that's a problem, okay? The telomere sequences, even the evolutionists would admit that it's very, very degenerate. And they kind of scratch their heads like, well, why is it so degenerate? Why aren't there more telomere sequences here? Because it's not a fusion. That's why. There shouldn't be any telomeres there because it's not two chromosomes that have fused together. Then they looked for that centromere, that other centromere that should be there, and they can't really find it. There's some sequences there that sometimes are associated with centromeres, but they're certainly not unique to centromeres. We find them in other places. So that's a problem as well. That other inactivated centromere isn't there. Now, there's many other lines of evidence that Dr. Tompkins has developed to show that it's just doesn't look like a fused chromosome. It doesn't match up with chromosomes 12 and 13. And one of them that he found, and he's published on this, and you'll see the reference in the next slide, is he found a gene there, okay, that spans the fusion site. Genes are always found on just one chromosome. I mean, it's not part of the genes here on this chromosome and part of the genes over here on this chromosome, okay? That doesn't happen. The gene's always on one chromosome. So how could it be that you somehow had parts of it on both and then they fused together and now those parts are together? That's just not realistic. So here's the supposed fusion site and here's the gene, right? Part of it's on one side and part of it's on the other side. The other thing is telomeres don't occur in the middle of genes. I mean, genes aren't around telomeres. So it's just filled with a lot of problems. And the conclusion is that human chromosome 2 is not the result of a fusion. It just doesn't match up sequence-wise with chimp chromosomes 12 and 13. So therefore, it cannot support common ancestry for humans and chimps.
Jackson Reed assumes that humans and chimpanzees are related through common ancestry at all. But at the end of the day, this data here, I'll just present just a few, forms of evidence that completely destroy the idea that humans and chimpanzees are actually related through common ancestry. They'll say human, chimpanzee are genetically identical by 98 to 99%. And yet we now know that this is based tremendously off of preferential and selective treatment of data. In other words, cherry-picked data. What they're using is they're using sequences that they actually expect to be similar. They don't include non-aligned DNA, gaps, copy number variations, and size differences. So sure, based on cherry-picked data and selective treatment of data, they might be able to get 98 to 99% similarity. But when you actually include and acknowledge it all, such as these gaps or these copy number variations, this non-aligned DNA, then what you actually get is something more like 88% or even lower. And at the end of the day, that's over 400 million DNA differences that actually exist between the two species. And most or nearly all the secular research reports on this human chimp DNA similarity omit significant amounts of data that do not align with each other. And there's significant DNA sequence discontinuity between the two genomes. That evolution, bacteria to biologists, evolution cannot explain or even account for. At the end of the day, the reality is that humans are not apes, and they were actually created separately and uniquely in the image of God. And not only does the Bible prove this, but so does DNA evidence. We can look to the Y chromosome. This is just icing on the cake at this point, because Y chromosome DNA destroys human evolution. The extensively and remarkably different DNA sequence of the Y chromosomes between human and chimp presents a very, very serious and detrimental problem. It's destructive. It's actually a fatal blow for common ancestry, because given the extremely low level of recombination and DNA sequence diversity on the Y chromosome, the human evolution paradigm encounters a fatal blow, as I've said, and a very serious problem. This problem is that the human and chimp Y chromosomes should be considerably more similar to each other. If human evolution was true, they should be the most similar chromosomes in the genome because they are so stable. In other words, there's a small amount of variation. And why is this the case? Especially when we know that every single human being on the planet is extremely similar genetically. 0.5% difference max, I believe it is. And all male Y chromosomes on the planet, almost identical, but yet chimp to human Y chromosome, 70% dissimilar. There's just so much evidence that destroys this idea of human to chimp ancestry. More icing on the cake. Let's look at orphan genes, for example. Orphan genes, they're fully functional gene sequences that code for both a functional messenger RNA and a protein, and they lack similar counterparts in other organisms. Orphan genes that are specific to humans are powerful evidence against the idea that we evolved from apes. Why is this? Well, it's because these genes appear fully formed, uniquely integrated into the complexity of the human genomic network, and clearly functional. And of course, very important to numerous cellular processes. And there's no evidence for common ancestry with apes or any other creatures. So as usual, as has been demonstrated here, in this short time, in this short video, genetics once again proves biblical creation and destroys fish-to-fisherman evolution. This amazing category of genes are taxonomically restricted genes that are unique, absolutely unique to each type of organism. These genes appear suddenly without any trace of ancestry in apes or any other animal. They don't fit the hypothetical fairy tale of evolutionary theory, and that's what evolutionists like Jackson Wheat here do, is they do a lot of imagining. That type of large-scale evolution only happens and occurs in the imaginations of those that actually want to believe in it. Orphan genes are evidently part of the genomic landscape that is indicative of Each created kind and the explanation of orphan genes fits the biblical model of created kinds, as does all other genetic evidence. The orphan genes are especially needed for the uniqueness associated with each created kind, as described in the book of Genesis.
Aside from genetic predictions, evolution also makes fossil predictions.
And when those predictions fail, they are not held as evidence against evolution, they're just ignored completely. Looking at the fossil record, we can see that ocean life dies in the ocean and land life dies on land. That's how it works. So we would expect to see aquatic life in deep sea layers because they were dying there far before the flood occurred. And until the flood came, which wreaked havoc on life existing at that time, causing dinosaur graveyards and things like that. Remember, when things die on land, they get eaten or degrade rather fast. The odds of life dying to make the quantity of oil that we find on Earth today is best explained by the flood. It's also safe to say that the fossil record is not valid when we find evidence of discrepancies in the record. And guess what? We do. The upside-down geologic column in Pakistan is one such place. At the foot of the Karakoram Mountains in the Salt Range Formation, scientists have discovered fossilized plants and insects. From the evolutionary perspective, these remains belong to the upper part of the geological column. However, this formation lies beneath the Cambrian rock, which is dated back to over 400 million years. Discoveries such as these support the biblical version of the Earth's history, whereby the geological column is primarily a consequence of the global flood and its aftermath. It is no surprise that there is some order to the fossil record, such as sea creatures buried beneath land creatures, and mobile creatures such as birds near the top. But, since as the Salt Range Formation testifies, the ordered charts that adorn textbooks aren't always what they find in the field. Many results today have kicked evolution hard in the dirt. They have found complete stasis in the entire Pliocene era. We also know that the Ashley phosphate beds in North Carolina have found human bones and artifacts with dinosaurs together. Also remember that they also found modern-day birds dating back in the Cambrian rock layers which supposedly evolved from dinosaurs. It's a total mess. Also consider that many of the biggest dinosaurs, such as the long-necked sauropods like the Brachiosaurus, Titanosaurus, and Apatosaurus, would have eaten colossal amounts of vegetation. So why do we not find such abundance of these plants in the rocks containing these dinosaur fossils? Take for example the Morrison Formation in Montana. Even though this formation had yielded many dinosaur fossils, they're very scarce in vegetation. This phenomenon of missing vegetation doesn't just apply to dinosaurs or their rock layers. The Coconino Sandstone in the Grand Canyon has many animal trackways, but it's almost devoid of plants. These rocks tell us something profound about Earth's history. They suggest that these deposits laid down are not ecosystems buried over eons of time. Otherwise, we'd find more evidence as plants that the animals ate. Instead, the evidence fits nicely with the biblical model of early Earth's history, whereas these animals were transported and buried catastrophically during Noah's flood. Geologists have assumed and say that calm weather conditions are required for these settling processes to occur to form mudstone. Therefore, whenever they encounter mud deposits, they interpret them as forming in tranquil water environments. However, recent research published in the prestigious Journal of Science has turned this argument on its head. The researchers showed that mud deposits form from rapidly flowing water. Yet again, evolutionary theory fails when put to the test, and we have more evidence for a global flood.
Darwin also observed that the bones inside the wings of birds looked like they were fingers that got fused together. So he predicted that a fossil of an ancient bird with separate fingers would be found one day. This ended up being Archaeopteryx.
You can find almost anything if you look in the fossil record that resembles something else. That means nothing. Paleontologist Lawrence Whitmer of Ohio University, in a commentary, stated, "Perhaps the time has come to finally accept that Archaeopteryx was just another small, feathered, bird-like theropod fluttering around in the Jurassic." What do evolutionists say about fully modern birds found in the early strata before Archaeopteryx supposedly evolved these fused fingers together? You guessed it, nothing. Stephen Jay Gould said, "It's a curious mosaic, not a transitional fossil." Even the stinkbird, alive today, is a species of tropical bird found in swamps, forests, and mangroves in the Amazon and in South America. It is notable for having chicks that possess claws. The truth? Hand claws are actually widespread in modern birds, and not all that unusual. Ornithologists say Archaeopteryx is a bird, as where paleontologists say it's a dinosaur. Not even a consensus between the sciences.
Another example of a very precise prediction concerns our yolk. However, eutherian mammals like us have a placenta that made the yolk sac obsolete.
The yolk sac is not obsolete. Think about it. If the yolk sac is a worthless remnant nowadays, like he says, then obviously it can be cut off from the human embryo because it isn't needed, right? Not at all. The yolk sac is the source of the human embryo's first blood cells and a blood nutrient supply. It's 100% required, as death would result without it. Lumen, the Boundless Anatomy and Physiology website, even tells us blood is conveyed in the walls of the sac by the primary aorta. After circulating through a wide-meshed capillary plexus, it is returned by the vitelline veins in the tubular heart of the embryo. This vitelline circulation absorbs nutrient material from the yolk sac that is conveyed to the embryo. It is true that the yolk sac of birds and reptiles contain yolk to nourish the embryo, but the placental mammal's yolk contains no yolk at all. Also, this so-called yolk sac is not a source of nourishment as it is for birds, or in a bird egg. Rather, its purpose and design is that of blood supply, proving it is not proof of evolutionary leftover remnants. To say that it's lost its primary function is intellectually dishonest. It's always had a function, and it was that of blood supply. Even Wikipedia acknowledges its importance by saying its function as the developmental circulatory system of the human embryo before internal circulation begins. Even embryologists today no longer call it a yolk sac, but umbilical vessel, because of its vital importance. This excerpt was taken from Larson's Human Embryology, 4th edition. The definitive yolk sac remains a major structure associated with the developing embryo through the 4th week and performs important early functions. Extraembryotic mesoderm forming in the outer layer of the yolk sac is a major site of hematopoiesis. Primordial germ cells can first be identified in humans in the wall of the yolk sac. Even by mistake or mutation, a human being cannot produce yolks or gills because we don't have and never had the DNA instructions for that. It's disingenuous to say that this proves evolution. Even Study.com states it was originally thought that human yolk sac was a vestigial organ, no longer of use to the embryo. But research over the past decade or so has brought new insight into the use of the yolk sac by the embryo. Also consider vitelline circulation, the system of blood flowing from the embryo to the yolk sac and back again. This system would not even exist at all without the yolk sac. This was never a useless, leftover remnants of a yolk sac. It was never a yolk sac at all, and should never have been called one. Hence, the modern-day name change. Do you see the lengths evolutionists have to go to present any evidence to support the lie? One can only wonder why evolutionists would call this structure by the wrong name and label it a primitive, when it's absolutely necessary for the survival of the developing individual.
However, eutherian mammals like us have a placenta that made the yolk sac obsolete. Paleontologists predicted an ant fossil that had features of wasps in the Cretaceous, and that was confirmed as Sphecomyrma.
For one thing, this species was never found to ever have any wings. No Sphecomyrma hamuli was ever found around the anterior portions. As were wasps, their fore and hind leg wings are hooked together with groups with tiny little hooks called hamuli, which ants do not have. Also consider, according to evolutionists, it was believed that this evolutionary age of 79 to 92 million years ago, there had not yet been any complex social organizations of ants into colonies. However, with careful analysis of these specimens, it reveals the presence of the metapleural gland, which is only found in ants, and only those that live in colonies. These glands secrete antibiotics which prevent bacteria and fungi entering the colonies. Ants were always ants. They never transitioned into flying, nor became wasps. Not a single drop of observational or testable science proves that ants became wasps.